Hello and welcome to another interview. This time you'll be interviewing Dr. Hugh Earl. Care to introduce yourself?
Yeah, sure, Brandon. My name is Hugh Earl. I'm a professor, associate professor in the Department of Plant Agriculture at the University of Guelph. Sometimes I'm the department chair. I'm taking a little break from that currently, and my area of research is crop physiology. So I study kind of sort of the interface between plant physiology and agronomy or crop production.
In one of your papers, you mentioned, while I was taking one of your classes, you mentioned quinoa research with Mr. James McCabe, possibly doctor now.
James was doing a master's degree. So yeah, he did his master's project on agronomy and physiology research in quinoa, that's right. So we had support from a private sector sponsor interested in quinoa production in Ontario, along with some money from the NSERC, the Natural Sciences and Engineering Research Council of Canada, and also matching funding from OMAFRA, the Ontario Ministry of Agriculture, Food and Rural Affairs. And between those three sources, we were able to complete 3 years of agronomy type research on quinoa at the Aurora Research Station, as well as some physiology studies to try to understand the crop a little bit better.
So what was the goal of this major project? And how were the group?
Sorry, my phone went off. I apologize. Could you repeat that?
So what was the goal of the project, and how were the room trials and field trials run? And what were the differences between these observations?
Okay, so we did, basically quinoa is a new crop in Ontario. It's only been grown on a very limited acreage. So there's very little information just on sort of basic agronomic practices. So just how to grow the crop successfully. So we were doing a number of very straightforward things, like looking at different row widths for planting the crop, different plant populations, different fertilizer approaches, so starter fertilizers and in-season N applications, that sort of thing. Other issues where, and we didn't really have a part of the project to address weed control, but that's a major issue with this crop. Basically, I know you know this, Brandon, but it's very closely related to lamb's quarters, which is a major weed in Ontario. So there aren't really any herbicides that are selective for lamb's quarters without killing quinoa. So there's weed issues. We weren't addressing those so much, but we also ran into some insect control issues, particularly with lygus bug. And so we actually, partway through the project, kind of diverted some of our resources to addressing that issue. But the other part of it, so that's just kind of your basic agronomy stuff, like how do you grow the crop? But when you've got a new crop, you want to do some work to kind of understand sort of how it works. Okay, so when you do agronomy research, well, you try this, you see what it does. You try that, you see what it does. But if you can actually understand the crop from a physiological perspective, like how does it form yield? When are the sort of critical times during that crop's developmental cycle where it needs to have, say, low abiotic stress, or is particularly sensitive to insects or whatever? If you're going to understand those underlying aspects of how the crop makes yield, then that can direct you, like, in a more directed, sort of focused way to try to develop the best possible agronomic practices. So one of our real objectives in this project was to look at quinoa, do a bunch of stuff to it to try to make it grow faster or slower at different times, and then identify those points in its development where it was most susceptible, right, to stresses that might then reduce its yield. So very basic physiological understanding of how the crop works in our environment.
So what differences were found between the indoor and field trials?
Okay, so yeah. So going back to this issue of when is it most important that the crop has ideal conditions? Okay, so let me describe how we approach that in a field trial. So as I said, we grow the crop in an experiment where we have like different seeding rates, right? Plant populations, different nitrogen rates, and we also had irrigation, right? To try to, and some of the plots, they grow better because they had water. And then what we do is, over the course of the season, we go and we just take some plants out, like we cut them off at ground level and we dry them down and we measure how much biomass is there. So if you do that enough times over the growth season, you can measure how the crop is accumulating biomass. Okay, so it goes in like a sigmoidal curve. Okay, so you get early on, the crop doesn't grow very fast, and it starts making a lot of biomass because it's got lots of leaf area, it's growing fast. And then at the end of the season, of course, it slows down again when it's maturing. So what we did is we looked at all these, I guess 40 plots, I think there were, and they all had slightly different growth patterns. And then we sort of correlated those growth patterns with, well, how did the final yield turn out? Okay, so it's kind of putting a lot of pieces together here. But the idea is that, maybe one crop was growing really, really well early in the season, but not so well kind of in the middle season and later. And then you say, okay, well, how did that plot yield? Maybe another one, early season wasn't so great, but mid-season it grew really fast. Well, how did that yield? And what we found with quinoa, the surprising thing, well, first, there was one thing that wasn't surprising, that high growth rates sort of right near the end of the growth cycle, those are important because that's when the seed is filling, right? So if the crop's growing really well late in the season, you get larger seed, which is good. That increases your yield. But the surprising thing was we were expecting that the most important growth stage would be right in the middle of flowering. So when the crop is flowering, and it's pollination's happening, and it's deciding how many seeds it's actually going to make, because that's the most important thing that affects yield in most grain crops, right? How many seeds are there? We expected that to be sort of right in the middle of flowering, as it is like for corn, for example. But what we discovered was that it was much, much earlier in the season than we thought it was going to be. So even just right before the flowers were even opening, just before you even saw any flowers in the crop, so it's very small, it hasn't even broken any buds yet, that turned out to be the most critical growth period to make sure you're going to get a high yield. So the seed number, which is the most important thing determining yield, was actually determined in this crop long before you even saw, well, not long before, but just slightly before you ever saw an open flower. Okay, so this was the really interesting thing that we found. And what we decided to do was to try to verify that by running a controlled environment study as well. Because in the field, you can kind of control when growth, what growth rates look like by different agronomic practices and stuff, but a lot of it's beyond your control. There's the weather and all these things happening you can't really regulate. But in a controlled environment conditions, and now we're talking about growing these plants in a room that's temperature controlled and humidity controlled, and it's got lighting is constant every day and all that stuff. So we basically grew the plants under those conditions. And what we would do is every now and again, we take some pots out and stick them under shade cloth. Okay, so what that did is it reduced their growth rate because they didn't get as much light. So we did this at different growth times. We take the plants out, put them under shade cloth, and put them back again under full light. And by doing that, we were able to slow their growth rate at different points in the developmental cycle. And what it did, this was James' master's work, both these things, the field study and the controlled environment study. What we found was the controlled environment study exactly verified what we saw in the field, that there was this critical developmental stage right before flowering when seed number was determined. And then later, very, very late in the growth cycle, just at sort of the mid to end seed filling stage, that's when the other critical stage was. But the early one was much more important, exactly as we saw in the field. So it's a way to take that, kind of surprising information you saw in the field and say, we really believe this? Like, how can we check this out, right? And so the controlled environment gave us a way to do that really objectively and not sort of being at the mercy of nature to test those ideas.
So do you know the physiological reason why resources and seed production happens is determined more so at that stage rather than at a strictly flowering stage?
No, honestly, we don't. I can only speculate because we're kind of making our measurements at the whole plant or the whole crop scale. And so why we got that result, if you want to talk about what's going on, say, in those flowering meristems of the plant, like of course, even though you can't see the flowers, we know there's already reproductive development going on, right, in the meristems. So presumably there's some decision that the plant is making that early on in the season that's going to affect how many flowers it actually produces. But what the physiological nature of that is, how that meristem is differentiating, or what maybe like plant growth regulator signals are involved, we just have no idea. We're just pretty confident that growth stage is really important. The crop has to be growing well, then if you're going to be able to maximize ideal yield potential.
Thank you. Now, during the experiment, in one of your field studies, lamb's quarters took over the field.
Yeah.
And that probably put a kibosh on at least one of your reps. How did that affect your experiment?
So we had a lot of agronomy trials, and they were kind of spread out over the research station. I'm talking in 2018 and 2019 as well. And some of the fields, unfortunately, did have a lot of lamb's quarters. So in our kind of physiology trial, the one I was just discussing, that was kind of there's a lot of resources in that trial. So we were able to justify going in with hose basically, and getting the weed pressure down to the point where it wouldn't affect our results. There are some of the agronomy trials in that year where lamb's quarters and also pigweed, it got to the point where we simply abandoned the trial. So we lost some of those agronomy trials, and we just had to discard those data. Like you just can't trust it, right, at a certain point. And after we did that, the first year of the physiology trial in 2018, we repeated it in 2019. And in that case, again, the weeds got really away from us. And despite our best effort, to try to do the right things up front to prevent the weed pressure. So we actually abandoned that trial in 2019. 2020 was when we were under heavy COVID restrictions. So we reduced our research effort in that year. And we didn't run the physiology trial that year. 2021, we extended the project just to complete that one trial. Okay, so we have those data from 2021. I haven't actually completed the analysis on that. Of course, I'm very hopeful it's going to look exactly like 2018, because then we'll really have it solved. But so that's kind of where that lies. The agronomy trials, some of them weeds got ahead of us, whatever, but we were able to get enough sort of repetitions of all the essential agronomy trials that I think in the end we've got a pretty good idea for some of these major issues. I think we have a really good handle on insect control, at least lygus bug. And I think we understand a lot about how the crop responds to nitrogen. And I'll tell you, the bottom line there is it kind of doesn't. It's another surprising thing about quinoa. In our hands at that location, if you applied lots of nitrogen to the crop, you can make it greener. So kind of mid-season, it would be darker green, and you can measure that with reflectance spectroscopy and those methods. But it never made any difference to yield. So as long as we had about 25 pounds of fertilizer nitrogen applied, nitrogen had no further effect on yield. It just affected crop color. So it's another kind of surprising thing. I think agronomy trials with this crop in other locations have found different things in terms of nitrogen response. But we saw it 3 years in a row with some trials that were really good. I mean, had like really low coefficients of variation, so very reliable trials. So another really surprising result. If you had just applied the nitrogen and looked at the crop, you'd say, oh yeah, it looks good. And if I didn't apply it, it looks kind of yellow. I'm not so happy with that. We never had a yield effect from nitrogen. And including in 2019, which is the year that we got the best yields. So we had yields approaching 3 tons per hectare, and so good yield potential, and still just not a response to nitrogen.
Wow. So what did you do with the lygus bug? What studies did you do to figure out how to deal with that?
So in 2018, we had a trial on, I think it was starter fertilizers. So we could see pretty early on, if any of those treatments were going to do anything. Because one of the response variables was just like, looking at early vigor of the crop. And we could see none of the starter fertilizers were doing anything. And that's the year we started to notice a lygus bug problem. So we said, okay, let's repurpose this trial and let's go and try some insecticide treatments. So we contacted Rebecca Hallett, who's an entomologist here at the university in the School of Environmental Sciences. And her group were great. They said, okay, yeah, we'll help. And they helped us apply some different insecticides and things. And they also took measurements of the actual insect populations. Okay, so they go through the crop. I don't know if you're familiar with this, Brennan, this method where they sweep the crop with these nets. They're called sweep nets. And they walk through the crop and they do this, and then they see what's in the net. And so they can count, lygus bug and whatever else is there. And they were able to show that several of the insecticides that they looked at actually did a really good job of controlling the insect. So, and then we repeated that in subsequent years, tried some different products and things. So there's a synthetic pyrethroid called Matador, which is a very common insecticide. It did a great job on controlling the populations and increasing yield. But I think Matador is actually scheduled for deregistration, I think. So that's an issue. Another one that we looked at is Saigon, and it worked extremely well. And I'm sorry, I don't know the chemistry. I'm not really an expert in that area. And then another one is a product called Closer, and it worked reasonably well, not quite as well as the other two. So from sort of just simply a chemical control standpoint, I think we know which products can control that insect in that crop. Of course, it'd be great to have non-chemical controls or sort of more integrated management type approaches, but that's not an area of research we were able to do in this project.
Thank you. Now, is there any other conclusions you would draw from this project other than what you've already said?
Yeah, so we're always interested in new crops, for Ontario, just to give producers kind of more options. So, for example, quinoa is mostly grown on contract, so it's possible to have a contract where what price you're going to be paid before you even grow the crop, and it seems like it has some advantages. For example, being productive on marginal grounds and that sort of thing. So maybe land that wouldn't be very high producing for our more traditional row crops. So it's possibly a good fit for Ontario potentially, but I would say at this point it's going to be a high management crop, just from the standpoint of not really having great weed control methods yet. And I know there's people working on that. I mean, there's very talented people working on solutions for that problem. And with any crop early on, I think there are there's going to be some teething pains, right? Some challenges to overcome. So I wouldn't be expecting thousands and thousands of acres of this crop in the province for a little while. But it does have some interesting properties, like this idea that you seem to be able to grow a pretty decent crop with no nitrogen fertilizer, even on a soil with only 3% organic matter. Like, that's pretty neat, right? So I think the crop definitely has some interesting properties that are deserving of further investigation.
Excellent. So if you could you go back in time and redo the project from scratch, what would you do differently this time around, knowing what you know now?
Probably we were really focused on sort of the agronomic practices, and we made some good guesses about the things that would work the best. I think if I had to do it again, I think I'd be interested in introducing more cultivars, more varieties of quinoa. We only worked with one, right? So the whole project was done with one that we knew was reasonably well adapted to Ontario because commercial crops of this variety have been grown. But I think because there's some data on different quinoa varieties in different parts of the province, but not really where we did our research. So I think if we had included maybe some more varieties, that would have been good information to have. Because it's not like one of these crops where we've got lots of varieties for each sort of maturity zone, and they all kind of yield about the same because it's so competitive. You know what I mean? Like, everybody's sort of optimizing. Very little breeding effort for this crop, certainly for our environment. So it would have been nice to know, well, is the variety we use kind of typical or the best? Or are there other ones out there that maybe could have done even better? So that's something I think I would approach in the future if I were to continue working with this crop.
Thank you. And now on to our last question. If you can give one last message to the entire audience, what would you say?
Who's your audience, Brandon, mostly?
Mostly it's homesteaders and such, gardeners and whatnot.
Yeah. So the message I would like to get out, and this is as a professor at the University of Guelph, and also a graduate. I mean, I did my undergraduate and my graduate degrees here at Guelph. The message I would give out is that agriculture in general and plant agriculture in particular are fields with endless opportunities for young people. And one of the biggest problems that honestly I think we have as an industry is a lack of awareness about the opportunities that exist either, you know, as researchers in agriculture, as I consider myself to be, or, you know, of course, people who are involved directly in crop production. But I think that being directly involved in crop production, being a grower, I think is, for some people, that's where the perception of the industry begins and ends. There's a massive industry in all of the different support industries that provide like inputs to growers, advice to growers, financial support, financial, you know, resources to growers, all of the different R&D areas in terms of plant breeding and pesticides, and just, you know, all of the things that support this sector. That's not going away, right? I mean, like the ag sector, no matter what happens to the economy, is always going to be important. There's always going to be work. There's always going to be opportunity. And I can tell you the major players and major employers in agriculture have a heck of a time recruiting talented people. There's multiple jobs for every single person who graduate from this institution with a degree related to plant sciences, applied plant sciences. So that would be my message. If you know young people who are interested in, you know, biology or any type of science or economics or any of those areas, agriculture is where you want to be. Really, there's so much opportunity.
Thank you. And I think that about covers everything. Thank you for watching.